Hello and welcome to Molecura. Today we will talk about mechanism of action of Oseltamivir and how it's going to disrupt the life cycle of the influenza virus. To understand the mechanism of Oseltamivir in a better way, we need to understand the basic structure and life cycle of the influenza viruses. So this is a structure of a typical influenza virus. We have a few outer proteins which we need to understand in order to understand the rest of the video. So the structures that are drawn here, the first one that we're going to be talking about is this surface protein, which is in blue color here. This is going to be our heme agglutinin. For the rest of the video, we're going to show this one as our HA protein. Now, the function of this heme agglutinin protein is binding to sialic acid receptors on the surface of the respiratory cells. The second one that we will talk about is the green structure over here, is a protein that we call neuraminidase. So let's try to write it down here. This is the neuraminidase. So the function of the neuraminidase protein is going to be cleaving the attachment of the surface protein, which we call the hemagglutinin, to the sialic acid receptor on the respiratory cell. In the next slide, when we are talking about the life cycle of the influenza virus, we will talk about this more. So the last surface protein that we're going to talk about is going to be the M2 protein. And the function of this M2 protein is forming a channel which is going to allow the influx of protons or H plus ions inside the endosome to release the virus's virion inside the cell, which we will talk about again in the next slide. So now we go to the M1 matrix protein, which is shown in red over here. This red color protein is the M1 matrix protein. So the function of this M1 protein is binding the surface proteins to the nucleocapsid, which is inside the viral cell. Now, when we go inside the virus, we are having the genome of the virus over here. This is going to be the genome. And let's try to draw the genome here as well. So this is the genome of the virus. So the genome of the influenza virus is going to be negative sense single-stranded RNA. And what does this mean? It means that the single-stranded RNA needs to be transcribed into a positive sense RNA before it can be translated to proteins. It is also important to note that this genome is segmented. So this is a segmented genome. And what we have is inside the viral envelope. And we have not shown it here as 8 pieces to make the diagram more tidy and simple. But there are 8 pieces of this genome inside the virus. So now that we have a basic understanding of the structure of the influenza virus, let's see how Oseltamivir is going to stop the spread of the virus within an individual. So what we have over here, this is our respiratory cell. This is going to be the cell membrane of the cell, and on top of this we have a sialic acid receptor. Here we have the nucleus, which has the endoplasmic reticulum attached to it. Now, what happens with the influenza virus is that we have our sialic acid receptor here. Let's try to write the names as well. Our sialic acid receptor. The influenza virus is going to come here, and there is going to be an attachment between this heme agglutinin surface protein to the sialic acid receptor on the surface of the respiratory cell. This is going to trigger the endocytosis of the virus inside the respiratory cell. So what happens, there is going to be an endocytosis happening here, and the virus is going to make its way inside the cell. Once inside the cell, the virus is going to be inside an endosome, which is covered by the lipid bilayer. So the virus is going to be inside this structure. At this point, the release of the virus genetic material is going to be mediated through the influx of protons. So there will be an influx of protons through this M2 channel, and this M2 channel is going to cause acidification of the endosome, and this will result in the release of the viral nucleocapsid and the viral genetic material inside the cell. So here we have our single-stranded RNA segments getting released inside the cytosol. And this is going to end up making its way to the nucleus. So this is going to be our nucleus, in which the virus is going to use the human cell machinery to replicate its genome. After that, there's going to be translation of viral proteins inside the human cells. And this translation of viral proteins inside the human cells is going to happen in two places. First, it's going to happen on the ribosomes inside the cytosol, and this is going to be for majority of the virus proteins. And the surface proteins, which are the hemagglutinin, which is going to be the M2 receptor, and the neuraminidase, is going to happen on the endoplasmic reticulum. So we are going to have translation of the virus proteins happening inside the endoplasmic reticulum. And what we end up having here is the viral surface proteins. So these surface proteins that are over here, along with the genome of the virus, is ready to be exported outside the cell to go and infect the next cell. So what happens for this surface protein is to get lined up over here on the plasma membrane. And once they are located on the surface of the plasma membrane, they are going to include the nucleocapsid of the virus within them and bud outside the cell. So the budding is going to look something like this. So there will be budding of the viral genome, which is going to happen from the cytoplasmic membrane with the viral surface proteins attached to it. So here the budding is happening. So the next step is going to be detachment of the virus from the cytoplasmic membrane. And this is the step that is going to be inhibited by our medication. So let's try to draw that here as well. So this sialic acid receptor is actually attached to the surface protein that we call hemagglutinin. So this is our hemagglutinin, which is shown in blue color. What we show here in the green color, as we talked about earlier, is our neuraminidase. So what happens is that this connection here needs to get cleaved, and this cleavage is going to happen through the neuraminidase. So what happens, the neuraminidase is going to come and it's going to cleave the connection between the hemagglutinin surface protein and the sialic acid receptor. So this is happening through the action of our neuraminidase. So neuraminidase is actually cleaving here. So here a cleavage is happening. So this cleavage is going to be necessary for this viral progeny to be released from this cell and to be able to go ahead and infect another cell. What happens here with our medication oseltamivir, it's going to come and it's going to physically block this neuraminidase receptor, so this cleavage does not happen. So the virus is going to be attached to this sialic acid receptor and it cannot go to infect another cell. So let's try to draw our oseltamivir over here. So this is our medication that we are talking about in this video. So this is our Oseltamivir, and you might know it as Tamiflu. So what it's doing over here, it's coming and inhibiting neuraminidase of the influenza virus and inhibiting the cleavage of the influenza virus from the sialic acid receptor on the surface of the respiratory cells. So let's try to write it down over here, what we just talked about. We have our oseltamivir. What it's doing, it's inhibiting neuraminidase on the influenza virus. It is going to inhibit the cleavage of hemagglutinin from the sialic acid receptor, and therefore it's going to inhibit the spread of the influenza virus inside the human body. Now, it's important to note that this medication is working by inhibiting the spread of the virus. So this is crucial for us to understand that all of these steps are happening anyway. So the virus is getting inside the cell through endocytosis. It's getting replicated inside the nucleus. The replicated virus RNA is getting translated to the surface proteins and other proteins inside the cytosol, and it's undergoing budding on the surface of the cell. So what is getting inhibited here is the release of the virus to the extracellular space. So what this medication is doing is stopping the ability of the virus to infect other human cells. It's important to understand that this medicine is going to be effective if started early. Once the virus has spread through the respiratory system, it is going to be of little benefit. Therefore, this medication is going to be effective only if started early in the disease. So effectiveness is to be started within 48 hours of onset of symptoms. This can be given as treatment for those who are already symptomatic, and it also can be given as prophylaxis for those who are in close contact with known cases of influenza but are not yet showing symptoms. If you are giving it as a treatment, is usually going to be a 5 days dosage twice a day, but for prophylaxis is just once a day, and it can be extended up to 10 days of the treatment. Now, before we conclude this video, we're going to be talk about another medication which is going to be inhibiting the M2 channels, which we talked about here. So this is our M2 channels, and we said that this M2 channel is going to allow the influx of protons to cause acidification of the endosome and release of the virus particles inside the cell. This can also be inhibited through the action of a medication. So this can also be inhibited, and this inhibition is happening by a medication that we call amantadine. So amantadine is going to physically block this channel, this M2 protein channel, and it's going to inhibit the acidification of the endosomes and the release of the viral particles inside the cell. But it's not longer used in practice due to its toxicity and due to resistance of influenza A to this medication. To try to demonstrate it more here, if we are having this channel, M2 channel over here, which is going to be something like this. So this is the M2 channel, and it's going to be allowing the influx of protons. But what amantadine does, it's going to come and it's going to block this channel. And the blockage of this channel by amantadine is going to be inhibiting the function of this channel. And by this blockage, the influx of the protons inside the virus is not going to happen. And if the virus does not get the protons inside it, it will not be acidified, and these genomes will not get released inside the cell. Now, this is the second antiviral that we need to understand its mechanism when dealing with influenza virus. But as we said, it's not going to be widely used anymore. Now, to recap what we have talked about so far, we talked about the mechanism of action of Oseltamivir, also known as its brand Tamiflu. We said to understand the mechanism better, we need to understand the viral surface proteins first. So the surface proteins over here, we had heme agglutinin receptor, which we have showed here in blue. This is our heme agglutinin. We also had our neuraminidase over here, and also we had our M2 protein over here. What we said is that this hemagglutinin protein is going to come here and is going to attach to a sialic acid receptor on the surface of the respiratory cells. And this is going to trigger the endocytosis of the viral particle inside the cell. And what happens once this endocytosis happens is that the virus enters the cell through an endosome. And the influx of protons through the M2 receptor is going to cause acidification and the release of the viral genome inside the cell. The viral genome is going to be single-stranded RNA, and it is segmented. It actually has 8 segments of single-stranded RNA, which are negative sense. What we meant by negative sense is that this RNA cannot be readily translated. It needs to be converted into a positive sense RNA to be translated into the product. The replication of this RNA is going to happen inside the nucleus of the respiratory cells. And after the replication is done, they are going to get translated into the surface proteins inside the endoplasmic reticulum. The rest of the viral proteins are actually getting translated inside the cytosol and not inside the endoplasmic reticulum. So once the virus had its genome replicated and its proteins translated, a budding process is going to happen where these surface proteins are going to line up on the surface of the plasma membrane, and the genome is going to go within this budding process, and this virus will bud outside the cell. Once budding outside the cell is where our medication is going to come into action. So this virus is still getting attached to the sialic acid receptor on the surface of the cell. So what happens once the heme agglutinin, which is shown over here in blue, is getting attached to the sialic receptor? This connection needs to be cleaved by something. So this is the action of the neuraminidase on the surface of the influenza. And this protein is going to come here and it's going to cleave this connection between the heme agglutinin and the sialic acid receptor. And this process is getting inhibited by our medication, which is oseltamivir. Oseltamivir is going to come and it's going to bind to the neuraminidase on the surface of the influenza, and it's going to inhibit its action for releasing this viral particle by cleaving the connection between the hemagglutinin and the sialic acid receptors. Because of this mechanism of action, we need to understand that this medication, it's not inhibiting any of these steps. The virus is still able to bind to sialic acid. It still can get inside the cell, inside the endosome. The virus can still get acidified by the M2 receptor. It can still get released inside the cell. It can replicate its genome in the nucleus. It can translate its RNA into proteins inside the endoplasmic reticulum. But once it wants to leave the cell and go to the next cell to infect more cells, it needs this cleavage to happen, and this cleavage is getting inhibited by our oseltamivir. So the way that this medication is working is by reducing the duration and the severity of the symptoms. That's why it is crucial for this medication to be started within the first 48 hours of onset of symptoms. Otherwise, if more than 48 hours have passed and this virus has already infected many respiratory cells, this medication is going to be of a little value to the patient. So it needs to be started as early as possible, and this can be used as treatment or it can be used as prophylaxis on those who are in close contact with a patient who is diagnosed with influenza. By this, we have finished the mechanism of action of oseltamivir or Tamiflu. If you enjoyed this type of content, make sure to subscribe to Molecura's YouTube channel to be able to explore more of similar videos on our channel. Thank you.